Okay, 11 o'clock on the first Sunday of the month, which means it's time for Chai and Y at Prithvi Theatre. Thank you all for coming on a Sunday morning. And before we go on to today's talk and today's speaker, for those who are attending for the first time or watching us online for the first time, Chai and Y happens 3 times a month, max. On the first Sunday at Prithvi Theatre, the third Sunday at Ruparel College in Watunga. And if a month is the fifth Sunday, then we are live from a lab in TIFR. So just to give you a glimpse of what may be happening, the mid-April Chai and Y in Ruparel College will be on astronomy, given by Anindya Ganguly, and then May and June, these 2 months are our special summertime programs, which are hands-on. They're most likely happening in the cafe outside. And the first Sunday of May, we are going to be doing, or everybody in the audience is going to be doing, experiments using a smartphone. And we're going to tell you exactly how we will do it. We will try and use all the sensors on our phones to do little experiments. So that's going to happen the first of our programs for the summertime. And today, we are going to be entering the world of chemistry or biochemistry, as you call it, with Rajeshri. She is just about to finish her PhD in TIFR. And those who are regulars may remember she did one of these summer special experimental sessions outside in the cafe a few years ago. Rajeshri is from Bardhaman in West Bengal, and she did her BSc in chemistry from Presidency University in Kolkata, and then joined TIFR for an integrated PhD in chemistry in the area of, I believe it's called chemical biology and molecular imaging. And what she tries to do is to actually track molecules inside cells, molecules that are important that we know where they are, in hopefully living cells. So, you can always kill a cell and see what's going on, but you don't want to do that. You want to image things. And one of the things you really want to image are lipids, fat molecules or lipids. And that's what she's going to tell us today. So over to you. Now you can start screen sharing. Okay. So we just make sure that the online audience can see it. See. Online audience, please remember that the only way we can get your questions and comments is over the chat. So if you're watching us on YouTube, please put your questions and comments in the chat, and we will then look at them. So can somebody confirm that we are on YouTube as well? Okay. So you can minimize this. You can also get rid of this by saying that hide floating meeting controls. Okay. Okay. Okay, all yours.
Thank you. Hello, everyone. Welcome to today's Chai and Y session, which is on tracking fat. So today we'll be discussing about how we can track lipid molecules of our body inside our living cells, and why it is important, why we bother to track the lipids inside our body. Okay, let's start. So first,
you have to press a button.
Where?
Button.
Still not working. It's working.
Okay, just give it a minute.
I think it will work. It should work.
Okay, try the buttons. Use laser.
This is also not changing.
Your computer is stuck. Oh, brilliant.
No, it's not.
Yeah.
Yeah. Yeah. Okay. Okay, it's okay. And then it's not happening actually.
Click once with it. Okay, then move that thing somewhere else. So that be right on the move it downwards.
It's okay. Okay. Fine. I think it's okay.
Everything seems to have made it just worse.
Okay.
Wait, now there's no light.
This thing went off.
Wow. It's on. Yeah.
Yeah. Okay.
Okay.
Fine.
Okay. Good. Let's just keep it here.
So Zoom audience can't see, right?
Yeah. With us for a minute.
Okay. So our body has mostly different kinds of molecules. Okay. So mostly we have these four kinds of molecules. One is called carbohydrate, which is basically sugar kind of molecule. Protein, which does all the work inside our body, and lipids, which makes a protective layer of our cells. Okay. So these types of molecules we also intake with our foods. And also the majority of our body is made up of water. So for today's talk, we'll be mostly discussing about the molecules which are fat or lipid. I will mostly be using lipid, and these are the actually fat molecules of our body. So in our body, we take two kinds of fat during depending on what we eat. So one can be like this peanuts, vegetable oils, or some fish has fat molecules.
Sharing some.
Hmm?
You are sharing the whole. I mean, this is correct here, but the screen shared over there is not the.
Okay, let me share again. Okay. Now it's fine.
Just hang on, let me check on this.
Let's see. Oh, there is some problem. Yes.
So earlier, this is what was coming. Now that's there.
You can also, but it's why it's zoomed in.
Yes.
It's fine in the Zoom.
Wait. Just wait. Let us just make sure that the Zoom is YouTube is correct. Yeah, Zoom is fine. Yeah, it's fine. Okay, go ahead.
Yeah. So what we eat depends on some natural fats or some processed fat. So what are the difference between the natural fat? So natural fat has this unsaturated carbon, but when we cook the fat multiple times, it gets saturated, and which is not natural for our body. So this is how this both the kinds of fat we intake during the food we eat, depends on what we food eat. And the for the daily intake energy, almost 25 to 50% energy comes from fat. So nowadays people say that go fatless and start eating non-fat food. Actually, that's not correct. Fat is important for our body. So the natural fat we intake, that helps us to form some hormones. Yes.
Go ahead, please ask.
Yes. This one, right? So there is a double bond between carbons and there is a single bond. So there are two hydrogen extra. So that makes the molecule stiff or floppy. So when the fat get absorbed in our body, if that is fat or floppy, like rigid or floppy, depending on that, the function may vary. Okay. So These kind of hormone like aldosterone, cortisol, estrogen, progesterone like hormones get synthesized from the fat molecule. Apart from that, some fats are good. They in our adipose tissue, which we have in our body, they store fat for the as a food for like emergency. So how it works? When you are not eating for a prolonged period of time, the body gets energy from this deposited fat, and that can produce energy at that point of time. Now each of our body is made up of a single cells, and all of the cell has a protective layer, and that layer is made up of lipid molecule or the fat molecule. Okay, so today's talk will be mostly about this protective layer of the cell membrane. So how this cell membrane looks like? This cell membrane has a lipid bilayer. So what I mean by lipid bilayer? Lipid bilayer is a two layer. Okay, two layer of same lipid, similar lipids. So they have, you can see that there is a line. So this is the upper layer, this is the lower layer. So I'll describe in details. So these molecules are looks like this, which has a head part which loves water, which has a tail part which loves oil. Okay, एक part को पानी पसंद है, एक part को oil पसंद है. Okay, so as our body is mostly made up of water, so cell के बाहर भी water होता है, cell के अंदर भी water होता है. So how they gets arranged? So the head part which loves water, they always face the water part either inside or the outside, and the tails see facing each other. How? Let's look like this. If you see these two molecules, their heads are showing either outwards or inwards, and the legs are facing each other. This is how the cell membrane gets arranged. Okay. So just think about it. Our cell looks like a room, right? This room. So if you want to go out or come in, you have to always interact with the wall, right? So this is the protective layer of, like, this is a wall of the surrounding of the cell. So if some molecule has to enter the cell, it has to interact through the wall. Okay. Similarly, if you want to interact, go inside the room, you have to interact to the wall, either by the door or from breaking the wall or something like that, right? So suppose some molecules want to go inside. Okay. So how that happens? Some molecules, this is our cell membrane, just a depiction. Now some molecule comes closer to the cell, and the cell membrane will encapsulate it like a packaging. And it will pack the molecules and safely deliver the molecules inside the cell so that they can perform the work they are supposed to do. Clear? Now again, something has to send outside. Then also cell can pack the molecule from inside in a lipid vesicle and send it outside. So these are the typical function this this membrane, the cell membrane can do. It looks like a packaging box. Okay. So people have used this concept to deliver drugs. So how? Because these lipids are biocompatible. मतलब already हमारे body में है. So what they does? They took some drug or something which they want to send inside our body. So they encapsulate these drugs inside this lipid vesicle, okay, or the nanoparticles like structure, and send it to body. So the recent advancement we have always we all have encountered is the COVID-19 RNA vaccine. So what happened? The RNA vaccine was the RNAs were delivered to our body by putting them in a lipid vesicle and injected in our body, and so that it can get delivered to our different parts of the body. Okay, so we are not interested about doing this lipid stuff outside. We want to see the lipids inside of our body. How? So we are mostly interested to see how this signaling things happens in a cell. Clear? Now there are 10,000 kinds of signaling lipids present in our cell membrane. So cell membrane, there is a cell, there is an outer layer of the cell membrane. The cell membrane is made up of almost 10,000 kinds of lipid. Okay, like suppose there is a wall. The wall is made up of 10,000 kinds of bricks. Okay, now suppose they looks like red, green, blue. Just differentiate in small amount of chemical structure. Okay. Now just think it in this way. Some Humpty Dumptys are sitting on a wall, and हम अभी बस दो ही color use कर रहे हैं. So yellow, there is a two kind of lipid. One is yellow and one is red. So basically, what happens when some signal comes, not molecules. Okay, when molecules comes, they get encapsulated or transferred inside, or vice versa. But जब signal आता है, they gets there are proteins which buried inside this, मतलब बहुत सारे proteins है जो cell membrane में होता है, वो signal receive कर सकते हैं. But उन cases में ऐसे packaging types नहीं बनते. तो उन cases में क्या होता है? Either some lipids का concentration, the lipid concentration of a particular kind of lipid get increased. Okay, or what can happen? They can change in position. Like the position of this lipid, there are multiple kinds of lipid, right? So there is a change in position of this lipid so that they can, the cell will know what kind of signal to transfer. So there are two kinds of signal I am talking about, where if there is a signal, there is a increased amount of lipid. So the cell will know what kind of signal to transfer inside the cell. Okay. And the another kind can be that there is a change in position of this lipid, so that cell will know that there is a different signal. Now you have to do some different signal you have to pass on to the other parts of the cell. Yes.
In a typical cell, how many different kinds of these lipids are there?
Almost 10,000 kinds. Okay. So the question is how many kinds of lipids are present? So basically, apart from cholesterol, there are almost 10,000 kinds of lipids. These are called phospholipids. They are meant to be transfer signals for the cells.
All 10,000 are there in each cell?
Almost, but the depends. Like suppose there is a skin cell, particular kind of lipid will be higher. Suppose there is a blood cell, that particular kind will be smaller, but the other percentage can be go up. So depends on the function they play. Depending on that, the concentration can vary. Okay. So till here, everyone. Yeah.
So the membrane that we mentioned earlier of the cycle, the job was to transmit certain goods from outside to inside.
Yeah. So there. Both. So basically, so the question was the cells transfer molecule from outside to inside. This is the only job? No. They can also do multiple other kinds of stuff. They are the main like, so if you want to enter the room, you have to interact the wall. So there are other kinds of things, like there are like apart from transporting molecules from inside to outside or outside to inside, they can do transfer signal, they can transfer the signals. Also, what they can do, they can host different kinds of protein on it so that they can play further pathways. Okay. So in cases of disease condition like diabetes, Alzheimer, cancer, and heart disease, here people have find that there is an increased amount of particular kind of lipid. Okay. So if you, what we can achieve by seeing these lipids, one thing we can see that fundamental thing, what is happening, to understand what is happening, like as textbook, like what we also need to know before treating something or something, disease or something, we have to know what is the actual function. Okay. To know the actual function, we can also see, if we can see the lipids, we can see the actual signaling pathway. Also in the disease condition, where the amount of lipid can go up or misregulated, then also we can talk about it is a disease condition. Okay? We have two aims, to do a fundamental study and a diagnostic study. Till here, everyone is with me? Okay. Now, there are some challenges. The challenges is, there is a different kind, as I have said, there are multiple kind of lipid present in the cell membrane, right? So they are not very different from each other. They are just slight difference in their structure. So they are structurally very similar. So detecting this similar structure is very difficult. And there, as I have said, there are multiple kind of lipid, but I want to detect a single kind of lipid so that I can talk about that lipid. So for that, the amount became very low. So in a hall of so many people, I have to detect one, like that. Okay. And the problem is, we have to do it in a live cell because we want to study the fundamental processes happening inside our body. Yahan tak sabko clear hai? Okay. Now, till now, what we do for lipid detection, the conventional way what people follow is the lipid profiling. So many of us has gone for this lipid profiling test, what it does, it takes out blood and detects how much kind of lipid you have in your blood. But the problem is, you are taking the blood outside, then taking the lipid outside and detecting. So this is no more life. They cannot respond to the signals, right? They cannot change their position. They cannot increase or decrease, because they are dead. Okay. So how we can understand that? Suppose एक India Pakistan का match हो रहा है, and I haven't seen the match. In the morning, I just only see these pictures. Okay. Can we say what happened during the match, or who has won the match, or is there anything important happened, because everyone is talking about the match? So pictures give you some information but cannot tell you the whole story. So for that, what you need a video, right? यहाँ तक ठीक है? Sometimes picture can also give you some wrong information, like this. Obviously the batsman is not hitting the other person, right? So what we try to do is, we try to make movie how these lipids change their position or concentration inside a cell. तो अभी लिपिड को देखने के लिए हम एक टेक्निक यूज करते हैं, that is called fluorescence. So many of you will know about fluorescence, right? So what is fluorescence? Fluorescence is the process जहाँ पे एक डाई होता है, ठीक है, एक मॉलिक्यूल होता है जिसको हम बाहर से लाइट डाल के एक्साइट कर देंगे तो वो एक्साइटेड स्टेट पे जाता है. जब वो ग्राउंड स्टेट पे आता है, क्योंकि उसका एनर्जी मतलब छोड़ना होता है, क्योंकि नो वन वांट्स टू स्टे इन एक्साइटेड स्टेट, तो वो जब एनर्जी छोड़ता है, दैट कम्स डाउन एज अ light. Okay. So basically when it will come to the ground state, it will emit some light that we can detect. जैसे कि अभी बहुत सारे पार्टीज में लोग फ्लोरोसेंट शूज पहनते हैं, जैसे कि इफ यू सी दिस गाय, ही हैज पेंटेड हिमसेल्फ विथ फ्लोरोसेंट पेंट. सो इफ द पर्सन इन like dark room moves around the room, you can also spot the person, right? So this is why we use fluorescence as our technique. Now what we do, we make sensor. Sensor is like we can say it's like a tracking chip. Okay, we'll put the chip on the molecule we want to detect, and that we can detect. Okay, wherever it will go. So that I'll call my sensor as detective cap. So what is detective cap? Suppose there is a cap only fits in the head of this girl. Okay, now there is a bulb on the hat. Okay, what happens when somebody else from this girl wears the hat? The bulb will not glow. Okay, now only when this girl will wear the hat, it will glow. ठीक है, यहाँ तक क्लियर? तो हमारा जो सेंसर है वो सिर्फ जब एक पर्टिकुलर, बहुत सिलेक्टिव है, वो सिर्फ जब वो पर्टिकुलर लिपिड पे बाइंड करेगा तभी वो ग्लो करेगा. ठीक है, तो उसके लिए मैंने एक एक्सपेरिमेंट किया है आप लोगों के लिए. So what happens? The girl is already wearing the hat. Now I am removing the hat in this case. Then I will again put the hat on. So you'll see this is gone. Like this is fluorescence actually. So there is a UV lamp behind. That UV lamp gives the energy to go up, energy to take the molecules to the excited state. Okay. Again, if I put the hat back, see. Okay. Now see, it is coming back, right? So this is called turn on, turn off kind of sensing. Okay? There is a sensing which is like it's on or off. But there is a problem with this kind of sensing. What is this? Suppose there is a room, right? This in a dark room, these 6 people were playing. Now, if the hat is glowing, then only you will know this girl is inside. Okay? Suppose there is no light at all. After you are looking for a lot of time, but you are seeing there is no light coming up. So you cannot say this girl is not there or the cap is not there inside the room. Okay, you cannot distinguish between these two things. So to solve that problem, what we did, we make a sensor which is already has some bulb glowing. Okay, suppose this cap has already bulb which is glowing in green. Okay. Now, whenever somebody else will wear that hat or no one is wearing that hat, that will glow in green. But when this girl will wear, that will change to blue. Okay, clear? So that has an advantage. Now you can confirmly say that the cap is inside the room. Okay. So I'll show again. See, this is our sensor. So if I put the particular lipid of our interest, it should change the color. Okay. So see, I'm mixing the lipids. You'll see it will change its color. So can you see? Can you see a color change from green to blue? Yeah. Okay. Now for that, now as I have said, I have a sensor now which can selectively detect to the particular lipid which I want to detect. Okay. Now what we need is a camera. So for camera we use confocal. So this is the camera which we use to image lipid molecule inside living cells. So this is a TIFR confocal facility where we do all our experiments. Now the problem which we want to address by using our sensor is this. So the one of the major global problem is breast cancer. Okay, there is estimate of like 50% of Asian women get breast cancer during the age of 25 to 50. And every minute there is a 4 women getting diagnosed with the breast cancer. Now also in India, one of the largest cancer in 2025 will be breast cancer. So this is a huge problem, okay? But there is a hope too. The breast cancer is curable if you can detect that early, okay? So the problem is it's very difficult. Till now, it's very difficult to detect the early breast cancer. So if you can survive, clear? Now, people have done a lot of research and shown particular kind of lipid, the concentration of that lipid goes higher. Okay? इसके बाद से मेरा talk थोड़ा सा technical होगा. I'll repeat. Just make sure you understand so that you can understand the concept totally. ठीक है? So first we need to know that we want to detect the particular lipid. I'll call it as PIP3. Okay? So that will be how this PIP3 is present and gets synthesized in the membrane that I'll talk about. So this is our membrane, clear? It has a receptor. Receptor is a protein which binds to external molecule and send the signal inside. Okay? So there is something called growth factor. Growth factor मतलब कोई molecule आता है और सेल को सिग्नल देता है कि नाउ यू नीड टू ग्रो और डिवाइड, क्योंकि हम बचपन से लेके अभी तक जो भी हमारा हाइट बढ़ा, जो भी बढ़ा, इसके जो सेल्स डिवीजन हुए, सेल डिवीजन होता है, क्लियर? ठीक है, ये जो सिग्नल है इसको बोलते हैं ग्रोथ सिग्नल. ओके, ग्रोथ फैक्टर आया, इस रिसेप्टर को बाइंड किया, मैंने क्या किया, इनसाइड सेल के अंदर सिग्नल ट्रांसफर कर दिया. Now this signal, after transferring the signal, this activates a enzyme called PI3 kinase. ठीक है, नाम याद रखने की जरूरत नहीं है, एक एंजाइम था जिसको एक्टिवेट कर दिया. ठीक है, हमारे मेम्ब्रेन में एक लिपिड और एक लिपिड होता है जिसका नाम है PIP2. ठीक है, हम जो लिपिड डिटेक्ट करना चाहते हैं वो है PIP3. हमारे सेल मेम्ब्रेन में हर टाइम एक लिपिड होता है जिसका नाम है PIP2. अभी ये एक्टिवेटेड एंजाइम PIP2 को PIP3 में ले जाता है. तो क्या डिफरेंस है? PIP2 में 2 रेड बॉल था, PIP3 में 3 रेड बॉल. बेसिकली ये एंजाइम यहाँ पे और एक रेड बॉल ऐड करता है. ठीक है, यहाँ तक सबको क्लियर है? तो हमारे मेम्ब्रेन में अगर हम कुछ कर नहीं रहा है, वो सेल कुछ सिग्नल नहीं है, देन व्हाट हैपेंस, देयर इज ऑलमोस्ट नो PIP3. PIP3 नहीं होता है हमारे सेल में, PIP2 होता है. जब सेल को ग्रो करना होता है, डिवाइड होना होता है, तभी PIP2 से PIP3 बनता है. ठीक है, नाउ बाकी के जो प्रोटीन है वो आके ये PIP3 को बाइंड करता है और देन इट गिव्स सेल सर्वाइवल, प्रोलिफरेशन, सेल डिवीजन, ये सारे सिग्नल फिर ट्रांसफर करता है. ठीक है, बट दिस विल नॉट स्टे लॉन्गर. जो when the work is done, there is another enzyme will come and take the PIP3 molecule to PIP2 back to maintain the PIP2 and PIP3 level so that the cell is functioning normal, right? Because PIP3 का काम क्या है? डिवीजन करना. डिवीजन हो गया तो उसको चले जाना चाहिए. ठीक है, यहाँ तक सबको क्लियर है? ओके. Now the problem in breast cancer, almost 50% breast cancer has the mutation in this enzyme PI3 काइनेज एंजाइम जो PIP2 से PIP3 बनाता है, ठीक है। देन व्हाट हैपेंस, इसको बाहर से कोई ग्रोथ फैक्टर की जरूरत नहीं होता. सो विदाउट एनी एक्सटर्नल ग्रोथ फैक्टर एडिशन दिस इज ऑलरेडी एक्टिवेटेड, ठीक है. तो दिस हाइपर एक्टिवेटेड एंजाइम कन्वर्ट्स ऑल द PIP2 प्रेजेंट इन आवर मेम्ब्रेन टू PIP3. तो सेल को क्या लगेगा, देयर इज अ हायर अमाउंट ऑफ PIP3, नाउ यू नीड टू ग्रो, राइट, डिवाइड, proliferate. Then that is the starting point of tumor formation. Okay, so cell started dividing uncontrollably. So this is the early stages what happens in the breast cancer, almost 50% of the breast cancer. So now we make a sensor, okay, that can detect this PIP3 molecule. Clear? Now experimentally what we see in our lab, this is a normal cell, ठीक है. अभी ये कलर जो दिखा रहा है वो उसका मतलब है ब्लैक से लेके अगर मैं व्हाइट तक जाती हूँ, देयर इज अ लो अमाउंट ऑन PIP3, अगर वो व्हाइट में जाता है तो हाई अमाउंट ऑफ PIP3, ठीक है. नॉर्मल सेल में PIP3 नहीं होना चाहिए, सो वी आर नॉट सीइंग मच PIP3, ठीक है. नाउ इफ वी ऐड ग्रोथ फैक्टर फ्रॉम आउटसाइड, दैट व्हाट हैपन, इट कैन इंक्रीज द PIP3 इन द सेल मेम्ब्रेन, क्योंकि उसका काम ही होता है सेल मेम्ब्रेन में PIP3 सिंथेसाइज करना. यहाँ तक क्लियर है? तो नाउ व्हाट यू आर थिंकिंग, if there is a breast cancer cell, what should happen? Anyone? More PIP3. So without putting any growth factor, there should be more PIP3. So in breast cancer cell, when we put our sensor, we see more PIP3 on the cell membrane. ठीक है? So it is quite distinguishing from the normal cell to the growth factor stimulated cell to a breast cancer cell. Clear? Now, there are drugs which are FDA approved drugs, which has been given to the breast cancer patients, but they are being given to every breast cancer patient. Okay. Now, suppose how these drugs are, like these FDA approved drugs are synthesized or get approved. What they does is they block this enzyme. So there is this enzyme, because of this enzyme, PIP3 was getting synthesized more. So if you can block this enzyme, there will be no synthesis of PIP3. Okay? Now you can control the synthesis of PIP3 and the uncontrolled proliferation. Now the drug which has been approved by FDA is this. So this blocks this enzyme and there will be no PIP3 synthesis. Okay? But the problem with this drug is there is a lot of side effect using this therapy. But the problem also is this has been given to everyone without knowing they are going to respond to this PI3 kinase inhibitor or not, because there is no testing of PIP3 ban raha hai ki nahi till now. Okay, so what we can address by putting this sensor? So one thing is we can pre-identify the patients, because there is a lot of side effects. So we can pre-identify the patients जिनके सेल्स को लेके हम डिटेक्ट करके देख सकते हैं जिनमें PIP3 ज्यादा होगा, सिर्फ उन्हीं को हम लोग ये ट्रीटमेंट देंगे, PI3 काइनेज इनहिबिटर उन्हीं को देंगे जिनमें PIP3 का लेवल हाई होगा. जिनमें नहीं होगा उनको वो ट्रीटमेंट देके कोई फायदा नहीं है, क्योंकि उनका कैंसर किसी और वजह से. ठीक है. ऑल्सो वी कैन डू अ पोस्ट ट्रीटमेंट मॉनिटरिंग. व्हाट इज पोस्ट ट्रीटमेंट मॉनिटरिंग? मतलब इफ यू हैव अ कैंसर which is because of the enhancement of PIP3 and you are given a PI3 kinase inhibitor treatment. मतलब तुम्हें treatment दिया, you had been selected that okay, this person will respond to PI3 kinase inhibitor. Then they have given to PI3 kinase inhibitor. Now what should happen? The PIP3 level on the membrane should go down. Clear? So what we should see by using our sensor is this is the breast cancer cell where PIP3 was higher. And if we put the inhibitors which blocks the enzymes, this can also lower the amount of PIP3. That means your treatment is working. Clear? Till here, everyone understood this concept? Okay. Now, what? Now you will be seeing that you are also showing us picture, right? You are not showing us movie. So for that, this is the sensor we have added to normal cell. And when we add EGF, it's a growth factor, basically epidermal growth factor. You will see that after putting the EGF, the intensity in the plasma membrane goes up. You see that the mark are coming out. So again, thus in our body, every cell does not respond simultaneous at a time. So some part will react faster, some part will not react faster. But if you see, as it is a natural binder, it will generate PIP3, but as soon as the work is done, it should go down. So see, again the intensity has gone down to the what was initially, right? So we can also track the PIP3 in the live cell plasma membrane. Clear? So what we are trying to do, TIFR is collaborating with this CCAMP. It's a institute, Translational Institute under DBT. So what they do, they take up this lab molecule to make it a commercialized product. So we are collaborating with them. They are testing our sensor in actual patient cells, that if they can detect this breast cancer cell and distinguish between different patients, different kinds of breast cancer. So we are also patented this molecule recently, so that if it can be commercialized. Also, I have published this paper recently from our lab. If you want to know more about this research, you can go to this paper, because it's an open access. You can always access this. And this is my lab members, and this is my guide who, with her, I developed this project. And the rest of my lab mates also work on detection of different kinds of molecules in our body, specifically molecules like metal ions, like potassium, calcium, manganese, these kinds of molecules. So they play also different kinds of role in our body. So with that, I hope to contribute to make our world better. And with that, thank you all for joining my talk. And if you have any question, I would happy to answer the questions.
Good. We'd ask you for the short summary, which is what you've given.
Okay.
You can also tell us exactly how your sensor is built.
Okay. So basically, that will be more technical. So the cap part is made up of peptide. Peptide is a small part of protein, and the the bulb part is made up of fluorescent dye. So as I have said, we are using the fluorescent. So if okay, so basically the cap is made up of peptide, and the head is the light is made up of fluorescent dye. So that dye has a special property. So special property means it can sense the polarity. Polarity मतलब पानी से oil में जाएगा तो वो there is a polarity difference. So that can sense that polarity difference. तो जब वो पानी में होता है तो वो ग्रीन फ्लोरोसेंट देता है, जब वो लिपिड में होता है तो वो ब्लू फ्लोरोसेंट देता है. और वो लिपिड में तभी जाएगा क्योंकि वो जो पेप्टाइड हमने बनाया है कैप, वो सिर्फ एक लिपिड के लिए सिलेक्टिव है. क्लियर?
Yeah. Okay, let's see yeah, fine, we can take questions. Should we take a break right now or come back for questions, or maybe we'll take a few questions and we can come back and have more questions, of course. So yeah, a few quick questions we'll do, and we can just check if there are any online questions, etc. So basically, what exactly is the mechanism of? We'll take all the questions here. Just
okay. So chemically, okay. Let me come to the maybe the figure. So if you see, so this is the peptide. Okay, I have a fluorescent dye on top of it. When it will see PIP3 on the membrane, it will go, because the peptide is selective for the PIP3, it will go and bind to the PIP3 molecule. And it was initially in the cytoplasm, which is basically water. It goes from water to membrane. Membrane is basically lipid. When it comes closer to the lipid, it will change its emission. But when because for every this peptide, because of this peptide selectivity is only goes to the membrane when there is a PIP3. Basically, chemically, this is what it is. So there is an interaction of this head group. It has multiple negative charge, and the peptide has such a kind of positive charge positioning. It is only selectively binding to this PIP3 molecule.
Maybe yeah. We'll ask you to simplify that after chat for a bit.
Yeah.
Yeah, go ahead.
So that the patients, we have to detect whether there is an excess of that protein, which
Lipid.
yes, of lipids. But you said that the patients will have excess lipids. So why do you have to detect it?
So all kinds of breast cancer does not go via this pathway. Okay, there is 50% chance that this is because of the enhancement of PIP3. Okay, so there is another 50%. Agar tum unko, you are treating them for PIP3 lowering, they are not going to respond, right? But you are giving them the drug. The drug has some side effect. So you are giving them the drug and the side effect, but you are not treating them. That's how you can previously, before giving the drug, you can previously decide whom to give the drug. Okay.
Any particular lipid that you see more in the breast cancer, whether it's cholesterol, it's referol, or is that
okay,
higher mark of?
Yeah, in breast cancer, this PIP3 is higher, definitely, but not like cholesterol. So basically, cholesterol makes gives the cell structure. Majorly, it is not a but signaling lipid. Okay, it gives us the structure and the strength of the cell membrane. Okay.
The cholesterol is high, and the doctors
Okay.
prescribe cholesterol medications. So how would that
Okay,
your research
exactly.
show?
So I will come that because of basically what happens in the lipid deposition in the blood artery. Mostly what happens, it's because this cholesterol, if you take this saturated versus unsaturated fat, they don't get absorbed by the body because they are not natural. They majorly get deposited on the artery of our blood vessel. तो ऐसे समझते हैं कि एक रोड था जहाँ पे नॉर्मली अभी गाड़ियां जा रही थी. Now there is a deposition of kachra. So the passage gets smaller, but you have to same amount of blood you have to pass through, right? Then your heart has to pump more because same amount of blood to flow, the pressure gets increased. That will get increase and increase. The lipid because of this lipid deposition, these things happen in the heart based or artery based disease. Majorly that happens because of cholesterol, and you will see that LDL and HDL count. Basically these are lipoprotein. So that gets deposited or that can convert. But in a cellular level, that is in a tissue level. It's a blood tissue that get deposited. It's I would say it's 10-fold higher order. But in a cell, if you see a cell, there is a change, very subtle change in the lipid. But the subtle change is such that it gets converted to a bigger level, transferred to a bigger level. So people are doing research to detect this if there is a particular kind of lipid gets changed. That is what actually fundamental research is all about. What is the normal state? What all kind of lipid you have? And what are the change concentration or distribution of your lipid in a disease, particular disease condition. Like Alzheimer, people are very interested to know because the peptide, the oligomer peptide, which binds to the cell and does this Alzheimer, is because of the alteration in the lipid concentration around this peptide also change the effect. So that people also are studying. Yeah, sure.
Because the recent thing, the research, whatever they're showing, I'm not from outside. They're saying the pharmaceutical companies to promote their anti-cholesterol medications, there is a level of there's nothing like to worry about cholesterol, let it grow and all that. So the doctors are having the people who read a lot, there is a difference in their acceptance that high level cholesterol means you need to take medication, and some say it doesn't matter, you don't need to take medication. So anything that you can.
Okay, so basically I'm not expert in this, okay, I don't work in cholesterol, but still what I can see, anything excess, even water, if excess in your body, it is bad. So basically you have to maintain all the things in a maintained way. But yes, if you are taking lipid, you should take the lipids which are of natural form, like vegetable oils, peanuts, they are good for your health. But if you are eating pizza once in a while, it's okay. But yeah, but if you are taking it on a regular basis, then your cholesterol levels should go high and there are a lot of side effects, right?
Thank you
So,
so much.
thank you.
Okay, I'll tell you what, we'll take a question there and then maybe we should just break, we'll end the live stream and then of course you can have as much time as you want. We have to get her to simplify what we have said.
Okay.
So we'll do that, but we will, yeah, go ahead, we'll take one more question and then we'll end.
Yeah, sure.
Unrestricted growth of peptide triggered by a growth enzyme.
Yes.
So the growth enzyme
Okay.
is genetically present in the body.
Yes, they are normal. So there is a normal cell growth, right? Okay. The question is it the enzyme which is making PIP3, is it present in a normal cell or not, right? So as I have said, that in a normal cell, they are controlled. When the growth factor will come, then only this enzyme will get activated. Then they will make PIP3 from the PIP2 molecule. But when it is a cancerous cell, you don't need a stimulus from outside. It is already activated because there is a mutation in the enzyme. Then it does not need anything to start. It always start. So it will act on PIP2 and make more PIP3. It's uncontrollable, basically.
Okay, so thank you. All right. So on this note, I would suggest that we thank Rajshri for taking
Thank you.
us through this. And online audience, sorry, you do not get chai, but you can put in your questions and comments still on the chat. If we receive some other questions, we will give them to Rajshri and ask her to type out a reply to them. Out here, let's go have some chai and come back. I'm sure she has more stuff to tell us about, and we can also ask and get clarifications. Just a reminder, 2 weeks from now at Ruparel College, we will be looking at astronomy, and the first Sunday of May, we will be looking at smartphone-based experiments that we all can do together. So those are the next 2 chai and Wise coming up. You can send an email to outreach, outreach@pifr.res.in, or follow us on Facebook, Instagram, Twitter, etc., and get to know the updates of the programs that are happening. So stay tuned and keep watching. Thanks a lot. Okay, that was just for the online audience. You can stop the live stream and end the Zoom meeting. We will have some chai and come back, and then we can start asking you more questions.